Hello, everyone, and thank you for joining us today for our Wild Insights webinar sponsored by the Claude Moore Charitable Foundation. My name is Devin Simmons. I'm the STEM Lyceums Program Manager, and I will be the host for this evening. CEE, or the Center for Excellence in Education, was founded in 1983 by Joanne DeGennaro and the late Admiral H.G. Rickover, who is the father of the nuclear navy and civilian uses of nuclear power, with a mission to nurture high school and university scholars to careers of excellence and leadership in STEM. CEE's mission is to, like I said, nurture high school and university scholars to careers of excellence and leadership in science, technology, engineering, and mathematics, and encourages collaboration between and among scientific and technological leaders in the global community. We also provide 4 programs that you do not have to pay for, and I will be going over those. So first, we have the RSI, or the Research Science Institute, which is a highly competitive 6-week summer science and engineering program open to rising high school seniors. Juniors are eligible to apply as well. RSI is the first cost-free summer science and engineering program that combines on-campus coursework in scientific theory with off-campus work in science and technology research. Then we have the USA Bio Olympiad, the premier biology competition for high school students in the United States, provides the motivation, curricular resources, and skills training to take them beyond their classroom experience to the level of international competitiveness. Then CEE began administering the USABO in 2003. Every member of the USA Bio Olympiad team has medaled at the IBO. We also have the Teacher Enrichment Program, also known as TEP. And TEP provides opportunities for middle and high school teachers to connect with leading experts from industry and academia, explore cutting-edge research and development, and make meaningful professional links with direct benefits for you and your students. And then last but not least, we have the STEM Lyceums program, which is the newest initiative at CEE. It's a virtual club for high school students to engage in discussions and explorations of STEM concepts or career pathways. And next, I'm going to introduce our speaker, and I am pleased to have Dr. Rebecca Johnson. Dr. Rebecca Johnson is the Associate Director for Science and Chief Scientist at the National Museum of Natural History. She is responsible for overall leadership and administration of the museum's research and collections activities. She oversees 7 scientific departments comprising over 300 staff, the Laboratory of Analytical Biology, the Collections Program, Care Registration Digitization, Academic Services to Interns, Graduate Students, Fellows, Research Associates, and Collaborators, and Emeritus Scientists, High-Level Liaison, and hosting of resident federal agencies, numerous cross-cutting strategic science initiatives and field stations. Dr. Johnson is a wildlife forensic scientist and conservation geneticist who most recently served as the director of the Australian Museum Research Institute and chief scientist of the Australian Museum. As chief investigator of the Koala Genome Consortium, she has significantly advanced scientific knowledge and contributed to management recommendations for the iconic Australian species. Dr. Johnson has an honors degree from the University of Sydney and PhD from La Trobe University, Melbourne, in the field of molecular evolutionary genetics, and has worked as a molecular geneticist in Australia and the United States. Dr. Johnson also holds the titles of adjunct professor at the University of Sydney, conjoint professor at the University of NSW, and spent 4 months at Kyoto University in 2019 as a visiting professor. We are so thankful for Dr. Johnson to be our speaker today, and I'm going to let her take over so she can give you some wild insight. Dr. Johnson.
Thank you so much, Devin. It's really fun to be here, and I'm a little bit envious of some of those courses that you introduced earlier. I would have loved to do those as a young person. It's my pleasure to be with you here today. As you probably gathered from Devin's introduction, I'm from Australia, and I have the great honor and privilege of being the chief scientist at the National Museum of Natural History. And today I just thought I'd share a few of my own stories in science and things that I have done and the places that I have ended up. And I've given it this title here, which is the Accidental Wildlife Detective. And hopefully that's going to come clear a little bit soon. I will start way back 20 years ago when I first started work at the Australian Museum, and I was then a junior member of the staff. I was the manager of the genetics lab, and I got a phone call from the New South Wales Police, who are the state authorities in the state where the Australian Museum is. And they asked me if I would be able to assist them in identifying a blood sample from a non-human source. And they told me this story about where this sample came from, and they thought that it might have come from one of these birds up here on the screen. This is a sulphur-crested cockatoo. And if you've ever visited Australia, they're very common in urban areas. You might have seen them walking through parks. They're very big, they're quite large birds. And the story that the police officer told me was that they had some complaints made about a vehicle that was seen driving through a park and seemed to intentionally drive over a whole flock of these birds on this screen here. And the birds at the time were being fed by people in the park. So families would come there and feed these birds every day. So it was a very distressing incident because unfortunately the car hit a number of these birds and 25 of them ended up either being killed in the moment or had to be euthanized because they had really bad injuries. So it's kind of a shocking story to hear from a police officer early on in my job at the Australian Museum. And so they asked me, if we gave you a blood sample that we've taken from underneath the car that had the license plate that belongs to would you be able to tell us if it comes from a cockatoo? It was especially important because the person that was alleged to be involved said that they were not there. It had nothing to do with them. There must have been mistaken identity. And so my training is in genetics, and I had never been asked a question like this before, but I thought, I know how to extract DNA from this sample. I know how I would get the DNA and what piece of the DNA I would get. And at the Australian Museum, we had plenty of birds like this cockatoo in our collection. So I actually had a comparison that I could make to it. So this is why I call myself an accidental wildlife forensic scientist, because I never dreamed that this is a career path that I would go down before this phone call. So moving on, we did the analysis and did indeed find that the blood was from a sulphur-crested cockatoo. And this went all the way to court, and the person had initially pleaded they initially said they had nothing to do with it, as I mentioned earlier, but they did eventually plead guilty, and they were charged with aggravated cruelty and negligent driving while disqualified. And it turned out this obviously wasn't produced in court until later. This person didn't have a license because they had already hit a person and killed them with their car. So that's a pretty sobering story for me to start with, but for me as a young scientist, it was a really unexpected way of applying the science that I had studied for many years to do. And I really developed a passion for it. And so 20 years later, this is a field we call wildlife forensic science. And doing what I did back then, identifying that cockatoo blood, has been done on many, many samples that you see here on the screen, things that are either illegally fished, like the shark over on the side here, or this snake that unfortunately is often illegally caught, to even scraping things off aircraft and helping the airlines identify what birds they may have hit in flight and how they can better manage that risk. So this turned into a whole thing at the Australian Museum, and is still a really active program there today and very successful. And they work very, very closely with different government authorities, applying science right at the edge of where the information needs to be. Many years later, I found myself here in Washington, DC at the National Museum of Natural History with the biggest bird collection in the world. And you can see an image of it here. And I have the great honor of leading the science for this incredible organization. And if you have not been to visit us, please do come see us, and you'll be wowed by this amazing elephant in the center of the museum and all the amazing exhibits that we have. And behind the scenes of those exhibits is the world's largest museum collection. And most of it is not on display because this is this collection represents about 148 million specimens. So it is enormous. It takes up many, many, many floors of storage, but it's used every single day for research and to answer questions like that one that I talked about earlier with the cockatoo. So today I thought I would tell you about my journey and how science has completely dictated how I've lived my life and where I've ended up, and I've had the great fortune of going to some pretty fun places. So right over on the left-hand side is me as a 5-year-old, my first year of school, and I said earlier I was very envious of those courses that are offered. I had no idea what a scientist looked like. I had no idea what a scientist did, and it's just such an incredible privilege if you get the chance to sign up to those courses and get that opportunity. Because very early in my career, I did actually want to be a scientist very early, but I had no idea how to do that. So I turned to my other love, which was dancing. So for many years, I really wanted to be a dancer. And I finally let that go when I got old enough and went off to university where I studied science and ended up doing lots of really interesting things and with the core goal that how can I use science and how can I use the skills that I've used to make a difference. And I've ended up right here at the National Museum of Natural History, but I've also met some pretty amazing people along the way. So you might recognize this person here, David Attenborough. He's just turned 98 years old. Well, this is him when he turned 90. He came to visit us at the Australian Museum, and for anyone that loves the natural world, there's nothing more special than meeting someone like David Attenborough, who has spent his entire life communicating the wonder and why it's such a privilege to have the world that we have. So I've got two parts to my story. I started off with the cockatoo, so you can recognize this little guy here. And the next part is actually the science behind how we fight wildlife crime. And the second part, you will have heard Devin introduce me as working on koalas. I am going to tell you a little bit about koalas and the work that we've done there. So I mentioned that cockatoo story, and it's a terrible story. It's animal cruelty. It's someone unnecessarily being awful and taking lives that they should not have. But it's actually an example of a much bigger thing, which is wildlife crime. Wildlife crime is unfortunately global. It's tightly linked to organized crime. It's very organized. It takes things like these shark fins up here, or these elephant ivory tusks, or logs entire forests. It takes things on scale, so it has a massive impact on not only those species that are being targeted, but also on the ecosystem. And it's worth an enormous amount of money every year. It's estimated, these figures are now 10 years out of date, but it's thought that it might be up to $200 billion a year that this trade represents. So you can probably see why this is of interest to organized crime. Unfortunately, this is a crime that's also growing. It seems to be growing at multiple times the rate of the economy, which is astonishing and not a good sign for the environment. So what do we need? What is needed to help with wildlife crime? Well, to investigate it and to prosecute it, just like any other crimes that you would think of when you think of forensics, good science and forensic science in particular, which is where science meets the legal system, is absolutely critical for understanding what's going on and prosecuting if people are breaking the law. This first example that I wanted to share with you is a DNA-based toolkit that we identified for species identification of rhino horns. So you might wonder why we chose rhino. They're a very iconic species. They're unfortunately very highly trafficked, and they're also endangered in most cases. And you can see these are some horns that have been seized, suspected to be illegal, and rhinos are protected. And I'm just going to flash up this picture here. This is a very disturbing picture of what happens when these horns are taken. Rhino horns are just like fingernails, so you could actually cut them off without hurting the animal. But because these horns are so highly valued in organized crime, they often will kill the animal and dig the horn out as far as they can so they can get as much volume as possible. So it's a really hideous, confronting crime. And there's a lot of countries around the world that want tools to be able to identify which species are being poached and where are they being poached from. And so I just wanted to introduce you briefly to the rhinos of the world. There are actually 5 different species. You probably think, when you think of rhinos, you probably think about the ones on the left-hand side of the screen, which are the ones from Africa. And those are the white rhinos and the black rhinos. And you can see they have in the thousands of individuals in their populations, but those are not really large populations. So they're considered near threatened for the white rhino and critically endangered for the black rhino. There are also rhinos in Asia. So you can see over on the right-hand side, the 3 different species of Asian rhino, and also, unfortunately, how small the numbers of those populations are. Unfortunately, all of these are trafficked. Even the Sumatran rhino, you can see the tiny little horn that this individual has. They're still, unfortunately, desirable in the trade. So they're all protected. And so having rhino horn is no-go, illegal under any circumstances. And so whenever authorities find these things, often they're in little pieces like this one here. So it's very hard to know what it is, let alone what species it comes from. So we went ahead, this was led by one of my students at the time, Kyle Hewitt. He went ahead and developed a very quick and accurate genetic test where he would take a DNA extraction of that little piece of rhino horn, like as much as a fingernail clipping. He would extract the DNA from it and he would sequence a short piece of DNA that was able to fit into one of these boxes here. And he's created this tree. This is what we call a phylogenetic tree. So this is made up of DNA sequences. And you can see here all the white rhinos cluster together for this little piece. All the black ones do, the Sumatrans and the Indians and the Javans. We also used horse, and that's firstly because we know it's a distant relative. So it's a good what we call an outgroup. But also horse, when you have something that's worth this much money, you get a lot of substitution. So horse is also something that's commonly seen in the trade. Horse is not illegal, but it's very hard to know what you're working with until you do this test. So what this represents is actually the science behind doing these identifications. And as I mentioned, there's a lot of countries that want this test. There was 5 different countries that validated this test in their lab. So that made it fit for purpose for forensic identification. It's very robust. See how these all cluster together with a very high score. This means that this is a very reliable test. So if you had an unknown and it's slotted right in here with the white rhinos, you would reliably be able to say that it was from a white rhino. And it's already being used in casework in countries that see a lot of this trade. So this is these are the samples that I showed you earlier. This is what they are identified as when we apply that test. One seizure of these little chips had 50 horns from white rhino, 50 horns total. 6 of them are from black rhino, 44 from white rhino. This one's from a water buffalo, so that is not protected, so they can have that one back. Black rhino, unfortunately, Indian rhino, highly endangered. And this interesting carving here was something that was brought to us for identification. You might want to guess what this is. Horse. And it was actually sold for a lot of money on the basis that it was rhino. So buyer beware, I say. So moving on to the second part of my talk, which is around future-proofing the koala with science. And why do I say future-proofing? Well, that's because the koala is, even if you haven't been to Australia, you probably can recognize this animal. It's one of the most iconic species in the world. It's definitely one of the most iconic species in Australia. But unfortunately, koalas suffer all the threats. They live in areas where humans also like to live, so we cut down their habitat and we build houses. They live in areas where there's a lot of traffic. They're affected by roads. You can see here some animals that are on some trees that have been cut down. They suffer from wildfires when that's in their region, and they try and shelter in the hollows of the trees, but the fires are so intense these days that can also impact them. And also, based on their very specialized diet, they're highly susceptible to the changes that we're seeing with climate change. So as the planet warms, their food source is actually getting less and less good for them. and I'll explain that in a minute. So that's why we chose to work on the koala, because it's a really important animal, and it's also very interesting. So you can see this girl here. This is a female koala in captivity. She's eating one of the only things that koalas eat, which is eucalyptus. They're highly specialized in the diet that they eat. And they eat the volume of eucalyptus that if we ate it, we would absolutely die. It has a lot of toxins in it that somehow the koala is able to metabolize or break down. And so that's basically all they eat. They also have this uncanny ability to find leaves that have more than 52% water in them. So that's also an incredible skill that they have. But I mentioned climate change earlier. As the climate warms and dries, then the leaves are actually getting drier. And so this is potentially quite impactful for an animal like the koala that has such a specialized diet. I wanted to tell you a little bit more about koalas. Not only have they got this specialized diet, they also give birth at 35 days to their marsupials. So they have a pouch, and their young is about the size of a jelly bean, this tiny little baby that crawls up into the pouch, latches on to the mum's teat, and nurses for the next 6 months until they pop out looking something like this little baby here. And so they do a ton of developing out of the body in the pouch. And so they have all of these interesting milk proteins where they are required to go through their developmental pathways. Now, I mentioned earlier that conservation is really important for this species, and that's because it is estimated that there's probably no more than 300,000 koalas remaining. It may even be half of that. This is a very difficult species to survey. And there are estimates that this might decline by up to 50% in some areas by 2036, and that's just over 10 years from now. And that's because of all of these threats that I mentioned earlier. They unfortunately lose their habitat to urbanization or to just agriculture and having trees cut down. They do suffer a form of chlamydia, and they can get it in their eyes, and it makes them blind. And so they obviously suffer a lot of threats if they can't see. They also get it in their urinary tract. And so that makes their bladder very fibrous, and it doesn't expand. And so they leak urine. And ultimately, unfortunately, they get cysts on their ovaries, and it makes them infertile. So it's a really many threats suffering these poor animals. And this is the distribution of koalas in Australia, this light blue range here. And if you've not visited Australia, if you've not seen a map, I wanted to give you a handy comparison here. Australia is a really big country. It's roughly the size of the United States. And so I've got Australia overlaid. So find your state here, and you can get an idea of roughly how big the two countries are compared. Koalas live all the way up north here, like right up to the Canadian border, right the way around into Texas. So they have a very big distribution, but it's very coastal. And so that means that they're very limited in the locations where they can live. And so we went about sequencing the koala genome as a way of facilitating conservation in this species. But what does that mean? What does it mean when you sequence a genome? How does that help with conservation? Well, firstly, I mentioned that the koala have this very specialized diet where they basically only eat a handful of species of eucalyptus. And the level of what we call plant secondary metabolites, which are these compounds that plants make to stop themselves from being eaten because they can't run away from predators. They have this very high level, but koalas have somehow managed to specialize in eating just that. So how do they do it? And so when we sequenced the genome, we did a whole big screen of what looked the same as other animals that don't have this amazing capability to eat eucalyptus, but what looked different. And we came up with this family of enzymes. And this family of enzymes, we all have them. We have these enzymes. You can see human sequences in here in red, the red dots. And basically anything that is dealing with compounds in the environment has these. So you'll see here what I've highlighted is that koalas seem to have a huge expansion in multiple places in these enzyme families. So and they seem to be functional. So what we think has happened is that over time, koalas managed to duplicate some of these genes, and they slightly modified their function to turn them into these super enzyme detoxes, so that they can detoxify themselves much faster than we could, for example, while they're eating this diet. So this is one interesting reason for sequencing a genome, where you can suddenly see at the DNA level how animals have these fascinating characteristics that help them survive. This is another full-on science figure that I'm going to talk you through a little bit. This is the other really important work that we thought was critical for part of the genome. Again, this is that distribution of koalas all up the east coast of Australia. And we took not only animals from the modern day, we also took some samples from 100 years ago, because obviously the habitat has been modified a lot in recent times. But unfortunately, when Europeans arrived in Australia, which is in the 1780s, they didn't take them long before they discovered the koala, and they hunted them very substantially. So we weren't really sure if hunting had also really impacted this population. And what we found was that across this very large area of the landscape, we found these 5 different groups of koalas that cover quite big areas. So you can see the red ones here, they go all the way down to here. The orange ones are kind of here, the yellow ones are down here, and then the different blues. And the main thing that I want you to take away from this is that these show big groupings of koalas that have had a lot of mixture over the years. And yes, they go between the groups, but there's these 5 big persistent populations that have existed over a lot of time. And in the meantime, there's been roads, there's been houses built. And so it's really important to maintain these big populations for the continued health of this species. The other thing that is really important to note is that some of these groups go across state lines. And like many things that you'll be familiar with here, things tend to be managed at the state level. So the koala doesn't know if it's in Queensland or in New South Wales, but the population is right on that boundary. So this is another key piece of information for us to share with the decision makers, that the koalas need to be able to go across these boundaries, because they don't know. So they don't know if the rules are different in Queensland compared to New South Wales. So this is another really important takeaway from this work. And another example, so I mentioned that koalas do get chlamydia. One of our collaborators on that work, that was his big focus, was developing a vaccine for the koala based on information that we learned from the genome work. And you can see the date on here. This is about a year ago, this publication came out. They had just started vaccinating koalas for chlamydia because it's such a debilitating disease. This is a really amazing outcome from this science, where these animals are now being hopefully being prevented from getting chlamydia due to this vaccine. It's not a silver bullet, because catching the koalas and vaccinating them is a lot of work, but it's a really great improvement on what was previously done, which was treating the animals once they had it with a lot of antibiotics, which they never responded particularly well to. So this is a really great example of science plus koalas equals better outcomes. And down the bottom here are actually a couple of very senior politicians from Australia, from the state and the federal government, who had both committed funding to do genome sequencing of all the koalas in their regions. And so that's, again, a really great outcome, where improved science, improved information is helping us to better manage this important species. So I'm just going to close by the way that I started, that I never thought that I would end up in the place that I've ended up as part of my science journey. But if you are thinking about going into a science career or studying science, no matter what you do, it opens your mind up to incredible opportunities in just different ways of thinking of things. You get to meet some of the most amazing people and work with some of the most amazing people, and probably do some good and advance knowledge in the process. And I've just got some pictures here of, I had the privilege of actually standing next to koalas on the stage, which is pretty fun. And these are some of the amazing people that I've worked with who have gone on to have their own fantastic science careers as well. And these are some collaborators that I have in the Solomon Islands. And we still work together today where we're working with the local tribes in the Solomon Islands on understanding and conserving their own biodiversity and their own health and other outcomes that all tie in together. When you save nature, so many other things improve. So with that, I really want to thank you for listening, and it's been a privilege to be here.
All right. Thank you so much, Dr. Johnson. Everything that you said was so impactful and so wonderful. Especially when you got to koalas. Koalas is one of my favorite animals, oddly enough. So learning more about koalas is always going to be interesting to me. So just want to thank you again. And so now we're going to transition into our question and answer portion. I'm going to ask Dr. Johnson some questions that I have already written down or typed out. Any of the audience members have any questions? At the end of my about 3, 4, or 5 questions, you can enter in your questions into the question and answer chat, and then we can take it from there. So my first question is, I saw that you were a dancer and then you transferred over into the museum field. And I was just wondering, for a young student, how can they combine their two passions if one isn't necessarily so STEM related into the STEM field?
That's such a great question. I think it's never too late to focus on your passion. So like that's what happened for me. I very early on, the first career I ever wanted was to be a scientist and cure cancer. And then I decided I really wanted to be a dancer. And I guess I kind of did both in the way that you're describing, where my hero was Marie Curie. Again, someone that was alive 100 years before I was, but she was the only scientist that I could even imagine learning about, because I just had no idea how to access a scientist. So I would say come to museums, meet people like me or the amazing people that work here at the Natural History Museum, and learn more about what they do. And if it excites you, then read more about it and see if it might be something that's of interest. And never be afraid to pick up something. It's never too late to pick up something. It might be a little bit late for me to be a ballet dancer these days, so that might be out, but it's never too late to learn something new and shift fields.
Absolutely. I'm a firm believer in you never stop learning something. So I definitely do agree that it is never too late to pick up something. And if you haven't had a chance, audience members, to view and go to the National Museum of Natural History, I would definitely go. I've been there. I'm a DC resident, so I understand how important and how just wonderful it is. Not just that museum, but the museums surrounding it. So if you have an opportunity, please go. My next question is, in your opinion, what are the emerging trends or technologies in wildlife science that students should be aware of? And then a follow-up question to that is, how can students get involved in these cutting edge areas of research?
Those are excellent questions. What is emerging in everything that I do is data. So if you understand how to handle data, if you like working with data, if you like trialing different analytical techniques, then that is absolutely emerging. And the best way to get a job in this field, if you can handle data. One thing that I'm really seeing in the wildlife forensics field is very rapid analysis. So something that used to take days or weeks is now, there's portable DNA sequences that you can take in the field. So there's a lot of really quick turnaround, which is super exciting, because you're most likely going to get a result if you can do a test as close to the event as possible. So that would be my first opinion. What was the second part of your question?
Is how can students get involved in these cutting edge areas of research or trends?
So there are, I think there are a bunch of participatory science projects that are emerging. I would love to say that we have some at Natural History, but we don't quite yet. But I think, at least for us, a lot of our big challenges are around making our data useful for some of these big initiatives. So a lot of our data is so old, it predates GPS, which I know it might be hard to imagine, particularly for the people that are listening. But our data is very old, and so it's like hand drawn on maps that give like a part of a state and maybe a county name with a road and a river. And so actually translating that into something digital is really challenging, but also quite fun because you get to delve back into history and go through notebooks of these original collectors. So there's a whole field there, and there's a whole piece of work that we need to do in natural history to make our data GPS worthy, so it can actually slot into the kind of technology that has been developed for those with those kind of coordinates.
Absolutely. Thank you for the answer, and I hope everybody was listening, because you are needed in this work. And so my next couple of questions, and then I do see that question in the question and answer chat, is what are some of the ethical considerations that come into play in wildlife forensic science? I know you mentioned with was it the cuckoo bird and things of that nature. So what are some more of the ethical considerations that come into play?
Yeah, so people often say to me, the wildlife crime is so global, like it just seems impossible. Like, what could we do as an individual? And so there are plenty of ethical decisions that you could make. So if you're on holidays in an exotic place or in a country that you're not familiar with, and you see something like a snake in a bottle, in a liquor bottle or something, which unfortunately is something that we would see a lot, and you think, oh wow, that's so quirky and weird, and I might buy that as a souvenir, then because by doing that, you are giving that market, you're promoting that market, and another snake will go get shoved into a bottle. And at least in Australia, if you tried to come into the country with that, it would definitely be taken from you, because firstly, it's you wouldn't have a permit to bring that animal in, and potentially would be an endangered species, or it certainly would not be allowed into Australia. So you're very likely to lose it as well. So that's an ethical decision that you as an individual can make. I think at the other really important role of museums in this type of work is that, and something that people don't think about with museums, is that we do a lot of research, and we have these big collections that you don't really even see when you come to visit us and look at our halls. But those specimens were, in many cases, that cockatoo that I did my original case on was collected 100 years before I did any work on it. And so it had been there, it had been used by many, many different researchers. So there is while collecting animals is very challenging and requires many, many permits and permissions these days, once they're collected, they have many uses. And so there's a renewable aspect to a museum collection where one animal unfortunately has died for whatever reason, and then it goes into our collection, and then it becomes incredibly valuable, so that it actually benefits its species by being in a collection, so others don't have to be collected.
Okay, that makes sense. And my next question is, following that same ethical question, do you know if there is like any rise or fall within poaching and hunting that is going on? I know that in the earlier 2000s, around when I was in high school, that was such a big problem that was going on. And especially, I think you mentioned the white rhinoceros. I can't remember if it's a black or white rhino that was close to extinction or was even extinct for a certain kind of amount of time. Is there any rise or any trends within that? And how does that affect your work when it comes to your wildlife forensic sciences?
Yeah, that's a really good question as well. So in answer to your rhino question, it was probably both species. So the white rhinoceros, there's 20, 30,000 of them, but they got down to a very low number. And it was thanks to a lot of captive breeding and a lot of attention on that population that got it back up in numbers. And so that's also a big use of resource to restore an animal that unfortunately that should never have been taken in the first place. So that's one thing. I would say apparently during the pandemic, there was a real rise in poaching in national parks, which is kind of, I guess it's not surprising, but it's really sad, because
Mhm.
so many people are out in national parks, and as like the only thing that you could do, it was enjoying nature, like a bit of a getting away from things. And so that was really unfortunate. The other thing that we see a lot of is there's a huge reptile trade. Reptiles are unfortunately traded a lot, and there's a big bird trade. And so those two things tend to, whichever country, those two things tend to be very prevalent. And
Mhm.
a country like Australia, it's one of the megadiverse countries, so it has a very interesting biodiversity as well, because it tends to be quite different to here, for example. And so a lot of even really common things like a lizard that you would see everywhere is unfortunately valuable in the trade. And so those are the two groups that you see traded a lot.
And how does that affect your research when you're at the museum or anything like that?
Yeah, that's also a really good question, because so that rhino test that I showed, that was an
Mhm.
entire year's worth of work. So,
Mhm.
so to get a test up to speed is it does it's not something that happens overnight. So you have to be
Right.
really nimble in what has been done in the literature. Has someone already worked on this species? Is there anything I can learn from what other scientists have done and transfer it into my work? So you often have to be very nimble. And then I have colleagues that work in some of the state labs or the federal labs, and they have species that they always see, like certain species of deer. So they have
Mhm.
really solid tests for deers where they can tell the gender, they can tell which population it came from, and so they can tell to the individual. And so it kind of depends on the amount of work, the type of work that you get. So in Australia, we got a lot of different stuff. So we're always scrambling. Is it a fish? Is it a lizard? Is it a mammal? So it definitely kept life interesting.
Absolutely, and I'm sure it will. And hopefully there will be changes within the population in terms of just the need for poaching and hunting. I can understand the delicate balance of it, but when it goes overboard, that's when it becomes a little bit sad to see, especially since these animals are just living their lives. So hopefully there will be a change in that trend.
Yeah, you
So that's
bet.
the end of that's the end of my question. We do have one question in the question and answer chat. Audience members, if you have any questions, this is your chance to ask, whether it's about anything with wildlife sciences, how to get internships within wildlife sciences or careers in that realm of speaking. But this question sort of goes back to what we were talking about with the rhinos. Why do they cut the rhino horn, and what purpose do they use for it?
That's a good question, and I should have mentioned that during my talk, but thank you for asking that. So I mentioned rhino horns are just like fingernails that you can cut them off.
Hmm.
They're even the same material. So
Hmm.
they're keratin. They're basically a very solid mass of keratin.
Right.
And unfortunately, there's a belief that rhino horn holds some kind of medicinal properties.
Hmm.
And this is where the one of the motivations comes from, that by powdering up the horn and drinking it, or by consuming the powder of the horn in some way, is going to make you healthier. You may as well chew your fingernails, I'll tell you that right now. And unfortunately, that belief got a big boost. A very prominent member of one of the Asian countries who claimed that he had cancer and it was cured from that. And so people not knowing any better, or not understanding the importance of, like, go investigate that, go find out what the actual chemical composition is, and would that be helpful for cancer? Unfortunately, a real escalation in the trade happened around that time when this very high profile individual claimed this. And so it doesn't cure cancer. I can definitely tell you that. But that is one of the big reasons why people take it. The other is that it has become such a valuable commodity that there's a certain status from just having it. And so it's like having a really fast car or something that you can show off to your friends. In some circles, having rhino horn has the same effect. Again, I would not be impressed, but unfortunately, some people find it impressive.
I don't think anybody here would be impressed with that, especially since, again, these animals are just living their lives, and so it is sad to see. Um, But that was the last question that we have in the question and answer chat. The person who asked that question just said thank you. And so if you have any more questions for Dr. Johnson, she is a wealth of knowledge. So please, after this webinar is over, if anything comes up, please email me and I can email Dr. Johnson, and both of us can learn something at the same time. So just thank you again, Dr. Johnson. Thank you for your insight and just your overall background. And so I just want to say thank you so much to our knowledgeable speaker once again. And another thank you to all of our sponsors, including the Claude Moore Charitable Foundation. And lastly, I want to thank the audience for joining us today. You will be notified when this recording is posted on our YouTube channel. More information regarding our next webinar will be sent out in the coming weeks, and we hope to see you there as we continue to grow within STEM Lyceums. Thank you very much and have a great rest of the day.